record my screen. Good morning, mga anak. We are now in the light-dependent reactions. Okay. As you can see, this is the illustration. This one is the thylakoid membrane. Remember the thylakoid? This one? Okay. If you have, if it is not clear to you, okay, please let me know. Okay, you can ask question. Ask the little bits of information so that I can also assist you, right? That is the membrane of the thylakoid. So if you're going to look at the thylakoid, let's have this. Okay. If this is your thylakoid, okay, this is your thylakoid, the membrane, okay, this one, okay, this is now the where the actual photosynthesis happens. The actual light-dependent reactions happens in the membrane of the thylakoid. So since it is a membrane, it is a phospholipid bilayer, okay? Okay. So this is now the layers, okay? And with the layers are your photosystems, okay? This is the layer, and in the layers are your photosystems and your ATP synthase. Now, what happens during the light-dependent reaction? Okay, during light-dependent reaction, photon strikes photosystem 2 first, or PS2 first, which contains your chlorophyll A, P680. Okay, once photon or a unit of light strikes photosystem 2, it will enable the electrons to be excited. Okay, and again travels to the electron transport chain. This one, this is the ETZ, electron transport chain. Okay, going to the photosystem 2. Okay, photosystem 1 rather. Once photon again strike photosystem 1, it will again excite the electrons and again converted ADP to ATP. This is now photophosphorylation. The conversion of ADP, okay, the adding of phosphorus to P, in the adding of phosphorus to ADP will make ADP into ATP. That is the energy currency of the cell, okay, and NADPH, okay. So there you go. This is now how we created, how plants created ATP. Again, how about the replenishment? How can we replenish the lost ions here? Remember that we lost electrons in the process because it is converted to ATP and NADPH, correct? Okay. How can we replenish the lost electrons? This is with the aid of photolysis, the splitting of water. This is the reason why plants needs water for photosynthesis. This is the reason behind, okay? Because water is really needed for the replenishment of electrons. When we split water, okay, we added electrons in photosystem 2, and again, oxygen is released as a byproduct, okay? Because this is a byproduct, when you say byproduct, okay, it is not needed as of the moment by the plant. It needs to be released in the atmosphere, okay? And hydrogen remains in the thylakoid gradient. This is now the gradient, okay? What is the purpose of having hydrogen ions in the thylakoid membrane? This is for us to create more ATP and more NADPH in the ATP synthase. It will be used by the ATP synthase to create more ATPs and more NADPH. NADPH stands for nicotinamide adenine dinucleotide phosphate. Okay, sure. What is that? Okay, how is that related to ATP? Later on, we will know that NADPH is converted to ATP again. Okay, this is actually convertible to ATP. How important is ATP? Well, that is the energy currency of the cell. we use ATP in our entire process, in our entire existence. We exist because of these ATPs. Okay? The raw materials for the light-dependent reaction is water, okay? Water and photon, your sunlight. Okay? And the products are ATP and NADPH. How many? Sure, it depends upon the plant. Okay? Counting now the ATPs created by the cycles will start, okay, in the light independent reaction, okay, going to the Krebs cycle, including glycolysis, right? Again, in the light dependent reaction, it started with photosystem 2. Light strikes photosystem 2, okay? But all of these processes are actually working simultaneously. This is simultaneous. But the process there is, it will strike again, it will strike first photosystem 2, going to photosystem 1. And of the creation of ATP and NADPH, the photophosphorylation. This is now the process of photophosphorylation. Okay? We lost electrons in the process. So what happens next is the photolysis, the splitting of water into hydrogen and oxygen. Once you split water, electrons will be replenished in the photosystem 2, and the cycle repeats again and again. Okay? And the hydrogen gradient in the thylakoid membrane, it will be added in the hydrogen gradient, and these hydrogen ions will go in the ATP synthase to create more ATPs and NADPH. Right? Do you have any questions so far?
None, sir.
Okay, please ask questions because these are just few illustrations and I actually make this as easy as possible. As you can see, those are just illustrations. You can always actually see the process there. Okay, even the arrows are there. So it is just really easy. The light independent reactions. Okay, let's move now to the light independent reactions. It happens to be coined as the dark reactions or the Calvin cycle. It was actually discovered by Melvin Calvin, right? Light independent reactions may occur is also occurring during daytime. It is not necessarily mean that light independent reactions occurs during nighttime. No, it it is also occurring during daytime. The only difference there is it is not actually using they are not actually using light. It's not using light as a raw material. There is no light involved there or photon. That is why it is called light independent. It is independent with the light. Okay, dark reactions or Calvin cycle. That is the other term for the light independent reactions. The light independent reactions of photosynthesis is a series of chemical reactions that produces glucose. Okay, this is the product, the C6H12O6 from carbon dioxide and other organic compounds. Okay, again, this is how plants created their own food. This is now the food, okay, the glucose. That is the product of photosynthesis. The ATP and NADPH, NADPH, okay, or nicotinamide adenine dinucleotide phosphate molecules produced in the light-dependent reactions, okay, are used in the light-independent reactions. So the product of light-dependent reaction, okay, the product of light-dependent reactions is the raw material for the light-independent reactions. Okay, again, the product of the light-dependent reactions will be the raw material for the light-independent reactions to occur, right? Producing a large organic molecules from carbon dioxide or CO2. The process involves a series of oxidation, reduction, reaction. Okay? There is an oxidation happening there, right? Passing electrons from protein to protein embedded in the thylakoid membrane of the chloroplast, forming glucose. Okay? As carbon dioxide diffuses into chloroplast from the atmosphere, the enzyme Ribulus 1,5-bisphosphate or biphosphate carboxylase or Rubisco. Yeah, this is your Rubisco. Okay? This is the enzyme that speeds up the combination of reaction of CO2 with 5-carbon carbohydrate. Okay? Later on, I will discuss it thoroughly. The Ribulus present in the stroma. Okay? Remember that the light-independent reaction happens in the stroma. Okay? This is the light-independent reactions. This is the entire layout. Okay? The Rubisco. Okay, Rubisco is Ribulus 1, okay, 5-bisphosphate carboxylase. So we coined that as Rubisco. So this is the Rubisco. Okay? We started with RUBP. Okay? Ribulus bisphosphate. Okay? Ribulus bisphosphate contains a 5-carbon molecule. It is actually a 5-carbon molecule. It is unstable. Okay? Since it is 5, okay, the Rubisco acts on it. Okay, this is the enzyme that fix carbon. The first phase of light-independent reaction is the carbon fixation. Okay? During carbon fixation, the carbon in the atmosphere, okay, the carbon in the atmosphere fuses with RUBP to create PGA or phosphoglycerate. Right. Supposed to be in a one phase or in a one cycle of um light independent reactions, we can actually create 3 carbons, this one. But in this phase, as you can see, this entire phase, this entire process, I actually created 2 cycles here. Okay? Because we can understand more if we will complete or we will create the complete cycles. We need 2 cycles of light independent reactions to create C6H12O6 or glucose. Okay, let's go back to the discussion. Again, your RUBP, ribulose bisphosphate, is a 5-carbon molecule or 5-carbon molecule. So because this is 5-carbon molecule, we need to fuse it with another carbon during the first phase of light independent reactions, which is the carbon fixation with the aid of Rubisco. Okay, once we paired in RUBP with 1 carbon molecule from the atmosphere, okay, we can actually create phosphoglycerate. Phosphoglycerate is a 3-carbon molecule. Okay, right? By the way, during the carbon fixation, carbon dioxide is being absorbed and used by the phosphoglycerates, and the oxygen is released again in the atmosphere. Okay. Sure. Why is it 12? Okay. 12 because your RUBPs, you have 6 RUBPs here. Okay. And again, paired in with 6 CO2, oxygen is being released in the atmosphere, and carbon is added in your ribulose bisphosphate. Okay. Remember that your PGA is a 3-carbon molecule. So therefore, we created how many PGAs? How many PGAs created in the process? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 PGAs. Because your PGA is a 12-carbon. It composed of 12 carbons. Or it composed of 3 carbons rather. So we created 12 PGAs in the process. Okay? Your PGA will be converted to pigal or phosphoglycerate. Okay? Phosphoglyceraldehyde. Okay? In the process, we actually what? Use 12 ATPs. Again, use 12 ATPs and converted it into ADP because the phosphorus there, we actually use the ATP in the process. And we use again the hydrogen ion in the NADPH or nicotinamide adenine dinucleotide phosphate. Okay, converting again the NADPH into a NAD plus. Okay, NADP plus. There you go. We have now 12 pigal or phosphoglyceraldehyde. Okay, your phosphoglyceraldehyde is a 12 is a 3 carbon molecule. Okay, so we have 36 carbons in the PGA and 36 carbons all in all in the pigal or phosphoglyceraldehyde. In the process again, we converted ATP into ADP because we use ATP in the process and we use also NADPH in the process. Okay. Your PGA, your pigal, okay, will be the one being used to create glucose. So we have 6. Glucose is a 6 carbon molecule. Because it is a 6 carbon molecule, your PGA, the 2 PGAs here, will be used in the creation of your glucose. And the rest will be the rest will go in the process. So since this is 12 minus 2, okay, minus 2 to create a glucose, 10 pigals will continue in the process. Okay, we have 10 phosphoglyceraldehyde. Okay, that is now the reduction phase. This is the second phase of the light-independent reactions. It is called the reduction phase. Okay, oxidation reduction. This is the reduction phase. In the reduction phase, we use 2 pigals, okay, or phosphoglyceraldehyde to create C6H12O6 or sugar or glucose. And the 10 pigals will continue the process. Okay. Next, we have 10 pigals for a total of 30 carbons. Okay. The 10 pigals will have a rearrangement of molecules. Remember, your pigals is a 3 carbon molecule, correct? They will rearrange themselves. And because of this rearrangement, we are using again ATP, 6 ATPs because these are 6 what? RUBPs. We have 6 RUBPs in the process. We are now again using 6 ATPs converted to ADP again in the process to create RUBP again. Remember the 30 carbons? Okay, your 10 pigals or phosphoglyceraldehyde has 30 carbons. Okay, and your RUBP, okay, has 5 carbon. It is a 5 carbon molecule. So in order for us to create a 6 RUBP, it needs to have a 30 carbons. Okay, 6 times 5 is 30. So again, we converted these 10 pigals into RUBP in the process. Okay, and we are using 6 ATPs to create 6 RUBPs. That is now the regeneration phase, the third phase of carbon fixation, the light-independent reactions. Again, the first phase is carbon fixation. We fix carbon dioxide. Okay, we fix carbon from the carbon dioxide. Okay, we need to join it with your RUBP to create a 6 carbon molecule. Okay, that's the first phase, carbon fixation. The second phase is reduction. Your 12 pigals, we use 2 PGAL or pigal in the process to create sugar or glucose. That is the second phase. That's the reduction phase. And the third phase is the regeneration phase, or the rearrangement of carbon molecules into RUBP. Again, this cycle supposed to be, it's 2. Okay, but again, I combined all the cycles in order for us to understand more about this process. Okay. Again, in the light independent reactions, we started with 6 RUBPs. Supposed to be it's 3 RUBPs. But again, because I added the entire cycle, the 2 cycles, so it became now 6. Okay? 6 RUBPs. So our 6 RUBPs needs also 6 CO2. The oxygen is eliminated in the process. Okay? And the carbon will be joining the RUBP through the help of the enzyme called Robisco. Ribulose bisphosphate carboxylase. Okay? It will be added in your RUBP to make your RUBPs a 6 carbon molecule. Because this is now 6 carbon molecule, during the process carboxylation or carbon fixation, right? Carbon joined with your RUBP to create 12 PGA, okay, or 12 phosphoglycerates. These 12 phosphoglycerates in the process will be converted into 12 phosphoglyceraldehyde. Okay, we are using 12 ATPs in the process, and we're using also 12 NADPH in the process because these are 12 PGAs. Okay, after which this 12 PGAL, we need to use the 2 PGALs in the process in order to create a glucose or C6H12O6. This is now the product of the light independent reactions. Okay, the 10 PGALs will continue the process, or the 10 phosphoglyceraldehyde will continue the process. 10 times 3 is equal to 30 carbons. This is 30 carbons. Okay, because this is 30 carbons, we can return this one to RUBP in the process of regeneration. This is the third phase of light independent reactions. We need 6 ATPs to create 6 RUBPs in the process. Okay, we rearrange the carbons to create ribulose bisphosphate. So there you go. We have now 6 RUBPs in the process. And the cycle repeats again and again. Okay, again, the raw materials for the light independent reaction is the NADPH, ATP, and CO2. CO2 can be actually absorbed from the environment. Okay, and the product is glucose or C6H12O6. Sure, where does the product came from? Take note. The product came from the CO2 in the environment. This one, 1, 2, 3, 4, 5, 6. Okay, expelled, the oxygen is being expelled, and the carbon continues the process in the light independent reactions. Do you have any questions so far?
None, sir.
Okay, don't worry. The illustration is in your module. You can download your module. You can actually see this one, right? I created this one in order for you to understand more. Other books don't have this. Even the books in other publishers don't have this. I created this one in my own. Okay, this is really clear. As you can see, this is the simplest version of all the illustration that I had been creating. This is the simplest of all. That is why I'm using this one. Again, the 3 phases of light independent reactions. We have carbon fixation by the help of the enzyme Robisco. We have reduction, the second phase, and the third phase is regeneration. Okay, rearrangement of carbon molecules to create RUBPs. All right. Question as of the moment? How about others? Jillian, do you have any question? EM?
No, at the moment, sir.
All right. Let's move now to the products of photosynthesis. Okay. The light-dependent reactions. Okay. Oxygen to water release as a byproduct. NADPH from NADP+. We added hydrogen ions there. Where does the hydrogen ions came from? It is the product of photolysis, the splitting of water. Okay. And the 2 electrons from the chlorophyll, ATP from ADP, and phosphate combined using the light captured or light energy captured by the chlorophyll. We are creating ATP, okay, NADPH, and the byproduct. Take note, it is a byproduct, oxygen. It should be released. It will not use in the process. Okay, so the products of photosynthesis is NADPH and ATP, which is actually the what? The raw materials for the light independent reactions. And again, the product of the light independent reactions is the glucose. It is a 6-carbon sugar, C6H12O6. Importance of photosynthesis. The sun is the primary source of energy of all the organisms on Earth. Primary source because there are some organisms that perform chemosynthesis. Okay? Scientists discovered that there are some organisms living in the seafloor, in the seamounts, okay, that perform chemosynthesis. Instead of performing photosynthesis, light is not present there. Okay? Therefore, they evolve. Okay? They actually adapt to the environment in order for them to survive. They are now performing chemosynthesis, the synthesis of chemicals found in the seamounts. Okay? However, the light energy from the sun can only be used when converted into chemical energy, your photon, a form that organisms can readily use in various functions. This made possible by photosynthesis. Okay? Organisms on Earth depend on the photosynthesis or on photosynthesis for survival. This is really true. In fact, if you're going to look at the ecosystem, okay, the pyramid of energy in the ecosystem, plants is in the lowest, but has the biggest energy compared to the other trophic levels. We call them as trophic levels. Okay, in the lowest trophic level, in the producers, autotrophs, that is the biggest because they are creating the biggest energy in the ecosystem. Okay, this is really true. And organisms on top is highly dependent on the producers or the autotrophs. The primary product of photosynthesis is glucose. Okay, your C6H12O6, a carbohydrate unit present in cellulose and starch, cellulose fibers and starch of your plants. Okay, glucose is used by plant cells to make organic compounds like fats, proteins, water-soluble sugars like maltose and sucrose. Maltose is from malt. Sucrose is your table sugar. Okay, plant need glucose for growth. Okay, on the other hand, animals need carbohydrates, fats, and proteins as sources of energy for various metabolic processes or cellular respiration. Photosynthesis provide an abundant or